Students, in the last class, we discussed about the microsporangium, that is the pollen sac. How the microsporangium is formed, the different layers of microsporangium and their functions. Today we will talk about the microsporogenesis and the development of the pollen grains. First of all, we must know what is microsporogenesis. The formation of the pollen grains from the microspore mother cell or the pollen mother cell is known as microsporogenesis. A microspore mother cell divides meiotically and forms a dyad microspores. The dyad microspore again undergoes a meiotic division and forms a tetrad microspores. The microspores or the pollen grains are attached to a common wall of the tetrad. Well, students, we earlier talked about the sporogenous tissues. A sporogenous cells called the microsporocytes may directly function as a microspore mother cell or undergo a few mitotic division to increase their number before they function as microspore mother cells. Now we come to the development of the pollen grain, a microspore developing into a full growth, a fully mature pollen grain. All of you must have seen a yellow powder on the flowers. All of you have seen flowers? Okay. Now these yellow powder, if you collect them and put them in a cavity slide with a drop of water and put it under a microscope, you can see amazing structures of the pollen grains. These amazing outer structures are of taxonomic significance. Now we shall learn about the structure of the pollen grain. The microspore represents the first cell of the male gametophyte. As the development continues, the four microspores of a tetrad become separated as individual microspore. Each microspore enlarges and undergoes mitotic division to form a large vegetative cell and a small spindle-shaped generative cell that floats in the cytoplasm of the vegetative cell. The vegetative cell has abundant reserve food and a large irregular shaped nucleus. The generative cell is spindle shaped with a dense cytoplasm and a prominent nucleus. At this stage, the structure is called as a pollen grain. Simultaneously, the spore develops a wall of two layers. The outer layer is called the exine, which is thick and hard and is made up of sporopollenin, one of the biological substances resistant to the action of enzymes, heat, and even strong acids. This is the reason why pollen grains of many species become well preserved as fossils. Exine exhibits a fascinating array of microsculpturing which is of taxonomic significance. Exine shows certain regions called the germ pores, where the exine is either very thin or absent. There are three germ pores in the pollen grain of dicot plants, while there is one in that of monocot plant. The inner layer is called the intine. The intine is thin and is comparable to the cellulosic cell wall of any other plant cell during germination of pollen grain. It grows out through one of the germ pores to form the pollen tube. Usually, the pollen grains are shed at two-cell stage in majority of the plants, while in some plants like maize, they are shed at the three-cell stage where the generative cell has divided to form two male gametes. The further development of the pollen grains occur on the stigma after pollination.